Imagine a cluttered laboratory, filled with petri dishes and test tubes, where the air is thick with the scent of chemicals and the hum of scientific equipment. A place where order and chaos coexist, creating an environment ripe for discovery. This was the domain of Alexander Fleming, a brilliant but somewhat messy Scottish scientist. Fleming's laboratory was legendary, not just for its disarray, but for the groundbreaking work that emerged from it. Colleagues often joked about its state of disarray, yet they respected his dedication and brilliance. But within this apparent chaos, Fleming possessed a keen eye for observation, noticing details others might overlook. Little did he know that his cluttered workspace would become the birthplace of one of medicine's greatest discoveries, penicillin. Fleming was a dedicated researcher, deeply engrossed in the study of bacteria, often working late into the night. His work focused on finding ways to combat the deadly infections that plagued humanity, a mission that drove him tirelessly. At the time, even a small cut could become infected, potentially leading to amputation or even death, a terrifying reality for many. Fleming tirelessly searched for a solution, driven by a deep sense of purpose and urgency. Little did he know, fate had a surprise in store, one that would come from an unexpected source. Fleming's keen interest in bacteria stemmed from his wartime experiences, where he saw the horrors of infected wounds. During World War I, he witnessed firsthand the devastating effects of infected wounds on soldiers, a sight that haunted him. This experience left an indelible mark on him, fueling his determination to find a way to prevent such suffering in the future, no matter the cost. His messy lab, therefore, was not a sign of negligence, but a testament to his relentless pursuit of a breakthrough, a place where magic and science intertwined. In the world of scientific discovery, chance encounters often play a pivotal role, leading to breakthroughs that change the course of history. Such was the case with Alexander Fleming's groundbreaking discovery of penicillin, a discovery that would revolutionize medicine. In 1928, while returning from a holiday, Fleming noticed something peculiar in one of his petri dishes. It was an anomaly that would change the course of medical history, an unexpected observation that would lead to a medical revolution. The petri dish contained a culture of Staphylococcus bacteria, a common cause of infections, which Fleming had been studying. To Fleming's surprise, a mould had contaminated the dish, forming a distinct ring around itself, an area where the bacteria seemed to be absent. What caught his eye was that the bacteria within this ring had been destroyed, while those further away continued to thrive, unaffected by the mould. Intrigued by this phenomenon, Fleming examined the mould under a microscope to understand what was happening. He identified it as a species of Penicillium, a common bread mould which was known but not fully understood in its potential. Fleming theorized that the mould was producing a substance that inhibited bacterial growth, a hypothesis that would need further testing. He named this substance penicillin, marking the beginning of a new era in antibacterial treatment. What seemed like a simple observation was, in fact, a momentous discovery that would save countless lives. Fleming's findings, though groundbreaking, were met with initial skepticism from his peers. The scientific community was slow to grasp the significance of his accidental discovery, doubting its practical applications. Many doubted the practicality of extracting and purifying penicillin, considering it a challenging and perhaps impossible task. Furthermore, the prevailing belief at the time was that boosting the body's natural defences was the key to fighting infections, not necessarily finding external agents like penicillin. This belief would soon be challenged by the undeniable efficacy of penicillin in treating bacterial infections. Fleming's initial attempts to extract and purify penicillin in sufficient quantities proved extremely challenging. The process was complex and yielded only very small amounts of the substance. Moreover, penicillin was unstable and broke down quickly, making it difficult to study and use effectively. Despite these setbacks, Fleming remained convinced of penicillin's immense potential. He published his findings in 1929, highlighting penicillin's remarkable ability to kill bacteria without harming human cells. However, his work received little attention from the medical community. The challenges of production and purification seemed insurmountable at the time. The world was on the brink of economic depression, and funding for scientific research was scarce. Fleming continued to experiment with penicillin throughout the 1930s, but he struggled to maintain its stability and produce it in larger quantities. His attempts to collaborate with chemists to overcome these hurdles proved unsuccessful. It seemed like penicillin's potential might remain unrealized. However, the story of penicillin was far from over. A decade after Fleming's initial discovery, two scientists at Oxford University, Howard Florey and Ernst Chain, stumbled upon his work on penicillin. Intrigued by the potential of this forgotten substance, they set out to continue what Fleming had started. Their journey was not just about scientific curiosity, but also a race against time. Their expertise in chemistry and pathology made them ideally suited for the task ahead. They meticulously planned their experiments, knowing the stakes were incredibly high. Florey, an Australian pathologist, was renowned for his meticulous approach to research. His leadership and vision were crucial in navigating the complexities of penicillin research. Chain, a brilliant German chemist, possessed an unparalleled understanding of organic molecules. His innovative thinking and deep knowledge of chemistry were invaluable. Together, they formed a formidable team determined to unlock the secrets of penicillin. Their collaboration was a perfect blend of scientific rigor and creative problem-solving. They recognized the urgent need for effective antibiotics, particularly in the shadow of World War II, where infected wounds claimed countless lives. The war added a sense of urgency to their mission, where infected wounds claimed countless lives. The battlefield conditions made the need for a reliable antibiotic even more critical. Working tirelessly in their laboratory, often late into the night, Florey and Chain devised innovative methods to purify and concentrate penicillin. Their relentless efforts and innovative techniques were groundbreaking. Their breakthrough came in 1940 when they successfully isolated penicillin in a stable form. This was a monumental achievement in the field of medicine. This achievement marked a turning point in the story of penicillin. It was a moment of triumph that would change the course of medical history. With a stable form of penicillin in hand, they were ready to take the next crucial steps. Florey and Chain could now proceed to the crucial stage of animal testing, a vital phase that would pave the way for human trials and ultimately save countless lives. Like and subscribe for more.